Hey everyone, it's very nice to see you virtually here. This is Fujian Lu from Boston Children's Hospital. Before I start, I want to thank the committee for the invitation to give this talk. I'm going to introduce our recent work on the establishment of cardiac diet and its calcium dynamics, the role of CMYA5. As the most important organ, heart keeps beating to refresh the circulation system till life ends. Heart beating or cardiomyocyte contraction relies on the very fundamental structural and functional unit, that is a diet. It is composed of two membrane systems. One is the T-tubule system that originates from the invaginations of plasma membrane into the cell interior. The other is sarcoplasmic reticulum, or SR, which is also fully connected, forming a network. When these two structures meet each other, they form dyads. So here is a typical EM micrograph of a cardiac dyad. It is T-tube here. This is junctional SR or SR. They are apart about 12 nanometers, forming the dyadic cleft. The T-tubule has L-type calcium channels, and the junctional SR is enriched with calcium release channels, RYR, in the membrane and the calcium binding protein, Ca2 in the lumen. During EC coupling, membrane depolarization opens L-type calcium channel and triggers a small amount of calcium influx. This calcium influx activates RYR to release larger amounts of calcium from SR through the calcium-induced calcium release mechanism. If all the dyads simultaneously release calcium, they will form a global calcium transit and induce cell contraction. Afterwards, the extra calcium will be cleared by the calcium pump in the SR, the calcium exchangers and pump in the plasma membrane, and the calcium uniporters in the mitochondria respectively. So as you can see, the intermolecular calcium signaling is largely constrained within this region. Dyadic proteins are extremely important for maintaining the architecture and function of cardiac dyads. Many mutations in them directly lead to cardiomyopathies, like many mutations in RYR can cause CPVT. So here, we'd like to use proteomic approaches to map the dyadic proteins and trying to dig out a set of regulators involved in establishing and maintaining the cardiac dyads, like the dyad formation, the junctional SR docking, the T-tubule maturation, and calcium signaling. And also, we want to study the dynamics of the proteome in different states like development and disease. To achieve the goals, we decided to use the BioID strategy. In this system, BA is a biotin ligase in the presence of ATP and biotin. It can biotinylate the adjacent proteins within around 10 nanometers radius. After strobed evidence beads pulled down, the samples can be analyzed by mass spec. To target the BA to the dyads, we fuse it to the N-terminus of dyadic proteins, junctin or triodine 1. To perform the in vivo proteomic mapping, we put the constructs into the adeno-associated virus backbone on the CTNT promoter for specific expression in cardiomyocytes. We injected the AAV into P1 pups at a full dose and waited until P21 biotin was continuously supplied for a week. At P28, we collected the hearts for strobe evidence pull down and then mass spec analysis. So immunostaining experiments confirmed the high transduction efficiency of our body sensors and also their correct localization. Western blot showed many biotinylation signals in the sensor plus biotin groups compared to the control groups. So after mass spec analysis, we overlapped the candidates from the two sensor plus biotin groups, excluded those also appeared in the control group, and then performed enrichment analysis. We found many classic dyadic proteins like RYR2 junctin, and so on, supporting the efficiency of our approach. Among the candidates, we found CMYA5 as a less studied protein. We confirmed its dyad localization by immuno-co-standing with RYR2 and also the proximity ligation assay. So collectively, the CMYA5 is a dyadic protein in close proximity to RYR2. So what kind of protein is CMYA5? Here is a historical review. Take-home messages, CMYA5 is a superlarge protein that is almost exclusively expressed in muscles. It can interact with a lot of proteins such as actinin 2 and desmin. It also appears to be restricted to junctional SR, where it co-localizes and directly interacts with RYR2, as validated by the immunostaining and also the co-IP assay. But what CMYA5 does and how it works remains a big question. To answer the question, we generated the CMYA5 knockout mouse line using the CRISPR-Cas9 technique. Briefly, two guide RNAs were designed to flank the largest exon 2 and consequently result into the early truncation at the very end terminus. We confirmed its deficiency at genome level, transcript level, and protein level. Then we started to characterize the phenotype. We first echoed the mice and measured the heart function, and we found that the knockouts had a weaker and weaker fractional shortening with the increase of age, while they have a larger heart weight, body weight ratio, and also a larger heart starting around 3 months. So this data suggests CMYA5 inactivation leads to a progressive cardiomyopathy. As the heart function is closely associated with the architecture of cardiac diet, so we set out to check the two membrane systems. We first took a look at the T-tubule system by loading the whole heart with a membrane dye. As you can see in the knockout hearts, the T-tubule system was severely disrupted as indicated by a significant decrease in the transverse fraction and a dramatic increase in the longitudinal fraction. To further ask whether the phenotype was the secondary effect of cardiomyopathy or CMYA5 functions in a cell autonomous way, we delivered two guide RNAs into the Cre-inducible Cas9 GLP transgenic mice at a high dose to validate the knockout efficiency and a low dose for mosaic knockout purpose. As you can see, the low dose AAV did not affect heart function, however, still disrupted the T-tubules in the GLP positive cells, while the T-tubules in the GLP negative cells remain quite normal. As also can be seen from the statistics, so this suggests CMYA5 cell autonomously regulates the T-tubule structure. So next, we went on to check the SR component. We scanned the circle and found that the patterns were quite similar to the Y type. Consistent with this, we expressed an SR-targeted fluorescent protein into the heart via the AAV-mediated gene transfer and found the gross morphology of SR actually was not changed. And in addition, the Z lines also appeared quite normal, as indicated by the marker actinin 2. However, we did find the junctional SR was severely disorganized, as can be seen from the mislocations of many dyadic proteins like RYR2, Ca2, junctionin 2, and FSD2. To see more details at the ultrastructure level, we did EM experiment and observed a quite abnormal architecture of the dyads in terms of the circularity of the T-tubule, the coupling ratio of the junctional SR and T-tubule, and also the distance between the dyads and Z lines, which are really important for the efficiency of EC coupling. The knockout hearts had a more variable and increased circularity of T-tubule and dyad to Z-line distance, as well as a decreased coupling ratio, suggesting that they may suffer from the defective EC coupling. So here we can say our proximity proteomics approach identifies CNY-A5 as a dyadic protein. CNY-A5 is required for the normal heart function, and also CNY-A5 deficiency disrupts the architecture of cardiac dyads. Next, we wanted to know how CNY-A5 regulates the architecture of dyads. Here we reviewed the establishment process of the dyads. One thing that we noticed was that the RYR2 moves to Z lines at a much earlier stage than the T tubules. As you can see here, at postnatal day 10, RYR2 is already well aligned to the Z lines. In contrast, the T tubules are still absent. Afterwards, their densities are becoming higher and higher till the cardiomyocytes are fully mature. So we isolated ventricular cardiomyocytes from P7 to P10 hubs. At this time point, we found the CNY-A5 and RYR2 were already co-localized at Z lines. It was also the same case for FSD2, which is an interaction partner of CNY-A5. In contrast, in the knockout cardiomyocytes, the RYR2 pattern was already disrupted, which means the CNY-A5 is required for SR docking to Z lines before T-tubule formation. Further, we wanted to know the docking order of CNY-A5 and RYR2, so we stand the E15.5 embryo heart sections with RYR2, CNY-A5, and actinin-2. At this stage, the Z-line marker actinin-2 was already regularly spaced. We observed 3 patterns. Some Z-lines only have actinin-2, like here. Some Z-lines have actinin-2, CNY-A5, but without RYR2. Some have like all of them. This data collectively suggests that there is a sequential docking of actinin-2, CNY-A5, RYR2, and T-tubules to the Z lines during the heart development. And also CNY-A5 is indispensable for the dyad assembly. But how does CNY-A5 localize itself to Z lines? To answer it, we again used the mosaic knockout strategy with guide RNAs targeting RYR2 and MYH6 respectively. We found that the localization of CNY-A5 needs sarcomere via the RYR2 is dispensable, since if we knocked the RYR2 out, the pattern of CNY-A5 still remained, while the MYH6 deficiency destroyed its Z-line pattern. So during the cardiac development, CNY-A5 positioning adjacent to the Z lines precedes junctional SR positioning or T-tubule formation, and CNY-A5 positioning requires sarcomere. As the architecture of dyad is critical for calcium signaling, next we studied the calcium signaling in the isolated cardiomyocytes. But before we go into details, I need to introduce the 2 new calcium imaging methods that we developed. So the first one is the dyad-targeted calcium sensor by targeting the genetically encoded calcium sensor GCAM6F to the N-terminus of junction or tridine 1, so that we could resolve the calcium dynamics at a single individual dyad level. The other one is the SR calcium imaging approach. Briefly, we overexpressed an esterase in the SR lumen so that it was able to induce calcium dye, in our case, Flu5N, into the SR lumen to directly visualize or monitor the SR calcium content here and also its dynamics. So we expressed the calcium sensors by AAV and firstly measured spontaneous calcium release events, and we found there were more calcium spikes in the knockout cardiomyocytes, especially on the isoproterenol treatment. Then we tested the cells to check the coordination behavior of the diets. We observed unsynchronized calcium release in the knockout cardiomyocytes as indicated by the curve instead of the relatively straight line during the initiation of the calcium transit. If we analyzed these diets one by one, we could see more details. So here is an example. As you can see, there are 3 passing events and 4 diets. So number 2 diet is well covered to the passing. However, the other 3, 1, 3, 4 missed 1 or more beats or had delayed activation. So on average, the uncoupling events were more frequent in the knockout cardiomyocytes. In addition, the SR calcium content was 25% lower than that in wild-type cardiomyocytes as measured by caffeine treatment. This was consistent with the decreased calcium transit amplitude and also the more frequent calcium release in the knockout cardiomyocytes. So finally, we put these animals into pressure overload condition by performing TAC surgery. So the knockout mice exhibit a much worse heart function, as you can see in terms of the fractional shortening, and with a much larger heart and also a higher heart weight, body weight ratio. It was also the same situation for heterozygous, although not so dramatic as knockouts. Also, we found a further damage in the knockout heart after TAC surgery in terms of the architecture of the cardiac diets as seen from the EM images and also indicated by the changes in the multiple parameters that are important for efficient EC coupling. Additionally, we observed a more disorganized RYR2 pattern, which contributed to the more aberrant calcium release in the knockout after the TAC surgery.
Moreover, we detected more fibrosis and also cell death after TAC surgery in the heterozygous and also knockouts. So taken together, the CMY5 deficiency disrupts diet architecture, its positioning at Z lines, and junctional SR calcium release, leading to cardiac dysfunction and inability to tolerate pressure overload.
Finally, we have the conclusion that the CMY5 establishes the cardiac diet or junctional SR architecture, positioning, and function. So the CMY5 deficiency leads to, at the molecular level, the mislocation of junctional proteins, especially RYR2. The ultrastructural level, the disrupted architecture of diets. At the single cardiomyocyte level is unsynchronized calcium activation and impaired the EC coupling. At the whole heart level, cardiomyopathy and intolerance to stress. Here, I'd like to give my acknowledgements to the group leader, Professor William Pu, for the guidance throughout the whole project, and my lab members and all the co-authors for their contributions. Also, I'd like to express my special thanks to the mentor for my PhD study, or maybe the whole life, the Professor Peace Chen, for being so much supportive. And the project is also supported by the American Heart Association Postdoctoral Fellowship. Thank you for your attention. I will be very glad to take any question you have.